సార్ కిషోర్ సార్.
సార్ చెప్పండి. హలో సార్. సార్ టీమ్స్
కాదు,
యూట్యూబ్ ఆన్ చేసేసేయండి సార్. బాపట్ల కాలేజ్ ఆఫ్ ఫార్మసీ యూట్యూబ్.
బాపట్ల BCOP@1995 స్టార్ట్ అయింది, చేసుకోవచ్చా?
సార్ మన కాలేజ్ యూట్యూబ్ ఛానల్ ఆన్ చేయండి.
నేను వస్తాను, ఇది అయిపోయింది.
మన కాలేజ్ యూట్యూబ్ ఛానల్ సార్ BCOP@1995.
నేను వస్తాను చెప్పండి.
సుధీర్ సార్ వస్తున్నారు లేపై.
ఇది సెట్ చేసి వస్తాను. చేయొచ్చా? రికార్డ్ పెడుతున్నాను. Sir.
Yeah.
Sir, I think it's ready, sir.
Yeah.
Sir, first two minutes madam మీ గురించి చెప్తారు సార్, ఆ తర్వాత మీరు ప్రెజెంటేషన్ స్టార్ట్ చేయొచ్చు సార్.
Sure. Yeah.
Okay.
Very good morning to all the participants and delegates. On behalf of Bavatla College of Pharmacy, I would like to introduce Dr. Bhaskar Rao sir. Sir has did his B.Pharm from Annamalai University in the year 1989. He graduated M.Pharm and PhD from Jadavpur University, India. Dr. Bhaskar Rao sir worked as a professor in various capacities in the field of pharmaceutical sciences in the reputed Indian universities. Dr. Rao sir has authored several research international publications and presented various symposiums and conferences. Currently, sir is working as regulatory director in New York-based clinical therapeutics. Prior to this, sir worked in various companies, BMS and Westward Pharmaceuticals, Om Labs and Perigo. Prior to industry, Bhaskar Rao sir has worked as research scientist for short tenure in highly renowned IICT Hyderabad, India. Currently, sir is working as a senior director regulatory affairs in US. So today is a great opportunity to avail as external speaker Bhaskar Rao sir.
Can I start now?
Sir, start sir.
Yeah. Hey, good morning and good evening to all the people who across the geographies. So it's a great opportunity. First of all, I would like to convey my sincere thanks to the respected principal of Bapatla College of Pharmacy, Dr. Gopal Krishnamurthy, who is showing the keen interest in the research activities individually as a college also. So that tempted me, but in spite of my busy schedule, but the kind of keen interest showed by the principal. So ultimately, I just took up this opportunity to give this topic of clinical regulatory and CMC regulatory. Purely, I'm going to talk about the industrial, physical, practical aspects. I also thank to the Bapatla College of Pharmacy Society and management and all the staff members and Rajalakshmi Garu for introducing me, and Sudhir, Professor Sudhir for arranging all the team activities, for arranging all this stuff. Thank you so much at the outset. It's a great opportunity. So I'm really thankful to all the society and also the respected principal who has given a lot of, the publications and patents in the international arena. So with this, I would like to, I know this is a huge topic covering around 30 days, but I wanted to complete everything within 1 hour and 15 minutes. So it's purely connecting, I'm going to mention the fundamentals versus the physical practical aspects in the industry, what is going on, connected to both clinical and CMC regulatory. Okay, I'm going to disclaimer is that the views in this presentation is completely the views of the other are mine from my personal experience and expertise in my personal capacity and does not reflect the views of the either the current or previous employers. It's a purely educational purpose and is only voluntary basis and is non-profitable. So this is a disclaimer. So straight away in the interest of time, I would like to go through the overview. First, I'll cover all the slides, so that you will have an idea. So in this presentation, I'm going to cover what is IND, investigational new drugs, before that preclinical and NDA, and then clinical protocol, clinical amendments, particularly in the phase 1, phase 2, phase 3, the branded companies which they follow typically NDA pathway, and chemistry and manufacturing controls. Typically, it covers under module 3 of ECTD, that is manufacturing sections. And there are two things. One is ICH, International Council for Harmonization. It's completely laid the complete guidance document for the entire whole regulatory agencies across the globe. And also I'm going to cover the pharmacovigilance, that is drug safety, from the preclinical and clinical phase 1, phase 2, phase 3 and NDA. It's called DSER. Development of safety update review, and periodic adverse drug reactions and periodic benefit. So this one, and of course I'm also going to cover the change controls. Change controls is one of the important factor which will regulatory impact assessment at each and every stage whenever you make any changes at every stage of phase 1, phase 2, phase 3. And also I am going to cover on the quality assurance impact assessment with respect to industrial scenario using the Viva Vault tools, which I am going to demonstrate in the a live example. So Viva tools is one of the best tools where all the industrial the departments will use extensively for medical affairs, change controls, and quality assurance, investigations, regulatory affairs, compilation of all the documents. I am also going to cover the type meetings, typically what we exchange, negotiate with FDA, right from the pre-NDA, IND meetings and everything. What are pre-IND meetings, end of phase 2 meetings, and pre-NDA meetings before NDA filing. So the sponsor will negotiate with FDA what type of changes, how the filing would be, either any lacking or something like what the requirements of the FDA. So at each and every stage, phase 1, the clinical phase of NDA, there is a continuous communication and dialogue with the regulatory agencies. Now I wanted to call that one as health authorities, So then I'm going to comparison of all the regulatory, agencies across the globe, different countries in the USA and EU. With this, I'm going to talk about, what is the regulatory landscape. Okay. First of all, I would like to show here, this is USA FDA. 1 second. So this is Health Canada is the regulatory agency is Health Canada, and US is mainly FDA, and all European EMEA and UK is MHRA, and of course Envisa is Brazil, and India is CDSCO, Australian Therapeutic Good Administration TGA. So likewise, each and every country across the globe, they have their own specific regulatory requirements. Mainly the strategy, how you're going to build up the strategy when you want to file in each country, as well as, each country has its own stringent requirements. Because once you file in FDA, even if you get approval from one product, it may not be accepted completely by other countries because other countries' requirements are completely different. So So then I'm going to show here one map, across the globe. Health Canada, FDA, Australia Therapeutic Good Administration, Brazil Envisa, Canada Health Products and like HC, European EMA, Likewise, Germany, France, but particularly in the Europe, there is a three type of submissions. One is mutual. When you file for Europe in one country, automatically it will be accepted by other countries. There is one more that is called centralized filing. The second one is mutual. So if one country is accepted the submission, the rest of the other countries also will be accepted. They will agree upon, and country specific also. And also there's one more called MENA region, Middle East nations. So they primarily follow the guidelines of the submissions. Once you file in UK or Europe, those submissions can be derived and they can be fine tuning to some extent, and they can be submitted into MENA, Middle East nations. So all these are regulatory landscape, the regulatory agencies across the globe. Likewise, China Food Administration, CFDA, Singapore HSCA, and Switzerland Swiss Medic, and UK MHRA, and of course US FDA. These are the regulatory agencies across the globe. The same document I'm going to show up here, you can also see it in the Google, all global regulatory agencies in the list. Then comes to the straight away I'm going to the topic. I'm going to cover, first of all, I would like to show all the presentation slides wherein you'll get an idea overall what I'm going to cover. Okay, this is the NDA pathway, preclinical toxicology, and what is IND, and then what are the ICH guidelines, which is very critical and important. Each and every regulatory country specific they will follow. Q means quality, S means safety, E means efficacy, and M multidisciplinary guidelines. And then I'm going to cover ICH is one of the document each and every country will follow, like module 1, 2, 3, 4. Okay. Then I'm going to cover what is the content of IND. This IND means that the content of IND submission documents for the phase 1, phase 2, phase 3, even for the NDA critical, more or less, all the NDA, including NDA, they follow the typical, the same, the modules, the contents. Just I'm going to show the layout, and then IND submissions phase 1 and phase 2. Then what are the module 1 requirements, particularly when targeting FDA? Form 1571, 1572, 3674. These are nothing but administrative documents, which I'm going to show, some point of time, what exactly it is. Okay. Then ICH M3 R2 guidance documents. Okay. It covers pharmacology, reproductive toxicology, genotoxic studies, general toxicology, toxicokinetics. So ICH guidelines, what are the safety guidelines? Then what are the efficacy guidelines? Efficacy, multidisciplinary guidelines, each one I'm going to talk overall. Of course, quality guidelines, CMC, drug substance and the drug product manufacturing. So how the QBD design, risk-based approach at a scale-up level, how the risk evaluation for the material attributes, process attributes and everything, how the risk evaluation, risk-based approach for the QBD. This is the typical diagram. These are quality guidelines. All these things will be covered in module 3. Likewise, what is the comparison between the USA and Europe? Comparison between the IND, NDA, biological applications, and 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 OTC, country specific. And then I'm going to talk about ICH guidance, all phase 1, phase 2, phase 3, phase 4, IND assessment teams in the FDA, what is the regulatory structure, NDA product quality assessment, CMC chemistry manufacturing controls module 3, what are the main type A, type B, type C, type D meetings, okay, with the FDA. Particularly I'm going to focus on the FDA. And then change management, change controls, Viva Vault, and what is the what do the regulatory say between the major impact, moderate impact, and minor post-approval changes? So CBE 30, annual report, prior approval supplement, comparison between the EU and pharmacovigilance, what is the SOSAR, SAE, SAR, AE, and everything, and of course clinical CRO functions, site how the clinical trials will be conducted, site initiation, site activation, patient enrollment, right? And then comparison between the China's CTA. The CTA is nothing but NDA, IND in USA. Other countries they call clinical trial applications. This is the one. So first of all, these are all these things I'm going to cover within 1 hour, in 15 minutes. So I mean, don't be panic. So I'm going to take, everything will be, very make it easy for you. How exactly the industry, the industry practice and the pharmaceutical industry works in a better way, So I'm going to show to you. So first of all, IND means investigational new drug applications. That starts from here. Before IND, any institution, research body, or research organization, or any company sponsor, they initiate a drug discovery called new molecular entity, new chemical entity. The synthesis of, for example, if any company wants to target any therapeutic areas like oncology, like respiratory diseases, like antidepressant drugs, So you wanted to choose a therapeutic areas, how to make more improvement in new novel drug discovery. Okay. So first of all, the synthesis of the API, active pharmaceutical ingredient, called initial synthesis. Once you do synthesis, animal testing, pre-IND called preclinical, where extensive screening will be done in the animal models. So rodents and non-rodents, like dogs, monkeys is one section, rats, mice, rabbits. So that depends upon how you are going to select the animal models. That depends upon the disease and the type of study what you are going to do. Okay, extensive screening will be done in the pre-IND preclinical phase. Mainly on the toxicology, LD50, like carcinogenicity, teratogenicity. Like I'm going to show it here. Preclinical toxicology is a critical component of the drug developmental process. The goal of toxicology testing is to support the transition of a new drug candidate to patients in the phase 1 clinical studies through the investigational filing. It mainly involves animal pharmacology and toxicological studies to assess the new drug. To what extent, what is the safe, what is the toxicological dose range studies. Dose range means from the low dose to high dose escalation studies, and also you derive what would be the best, the dose can be administered, to the human beings. That is in the phase, IND phase 1. Okay. Today I'm going to mainly cover in the small molecules. There are two things like small molecules means biologics. Biologics are bigger molecules, means that the peptides, insulin, like vaccines, all these are called as biotechnological products. They need a regulatory requirement is highly stringent, but more or less the same type of pathway, whatever you are going to discuss here. Likewise, the toxicological studies in the animals, they single dose toxicology, repeat dose, okay, and reproductive, developmental and genotoxicity. To what extent it's going to cause a mutation in the animals, okay? By appropriate analytical methods, they will evaluate, okay? So extensive screening will be done in the animal studies. So once you complete all the studies, gather the complete information, like animal toxicology and everything, like nitrosome and impurities, like impurity studies of the drug substance, whatever is coming, because once you manufacture the active pharmaceutical ingredient, it may contain degradants or impurities that may cause even nitrosome and impurities. Recently, that the FDA and worldwide there is a concern about the nitrosome and impurities. Even metformin, the old product, because of the the nitrosome and impurities, the FDA has issued the CEDAR guidance. Each and every molecule product which is going to come in the market newly, they also need to evaluate carcinogenicity and nitrosamine impurities because they are causing a cancer actually in the animals. So once you evaluate at the pre-IND phase in the animal studies, gather the first gather the complete literature and the data. Okay, then you set up a called pre-IND meeting. Pre-IND meeting with the FDA. Hey, this is the data what I am having, and also what is the proposal you are going to carry out, what is the dose. The first dose, F5H, first in humans dose will be conducted, in the IND phase. So from the IND phase onwards, phase 1, phase 2, phase 3, all the drugs should be manufactured under GMP conditions. GMP facilities, of course, it is a research bodies or companies or whatever are any sponsor, So from the IND onwards, the dose will be administered, it will be tested in the humans, phase 1, phase 2, phase 3. So if the agency says that, the data is not sufficient, they will ask you to do the protocol amendment. That time you need to do the change controls and what is the, again, modify the protocol and submit to the agency. If the agency says yes, then one can start the phase 1. Phase 1, phase 2, phase 3, what is the main difference is that in the phase 1, okay, whatever the recommended, the safest dose you already recommended from here, it will be administered to the small group of humans, maybe 20 people or 30 or whatever. So there is a protocol, it has to be approved by the agency, okay, and also it has to be approved by the IRB or IEC. IRB means that is called Institutional Review Board. IEC means Institutional Ethical Committee. Okay, once this is approved and everything, in the small group of human beings, we have to go do the studies like clinical. Mainly here also safety and phase 2 safety. Maybe the diseased human beings are healthy human beings, but when it comes to the phase 3, one has to completely evaluate in the larger group of human beings, subject, particularly on the diseased patients, not the healthy patients. But in the phase 1, phase 2, maybe diseased or maybe healthy. So in the phase 1, maybe 20 subjects, phase 2, maybe 50 or 100 subject, larger group. Of course, you are going to see the mainly tolerability within the dose range. What are the side effects you are going to encounter? Once you establish in the phase 2, okay, then it comes to the phase 3. Okay, in the phase 3, larger groups, maybe 10,000 or 5,000 or whatever. So here the main emphasis on the phase 3 is that efficacy. To what extent the drug is more effective in the treatment of the disease, right? So of course, at the same time, monitoring the tolerability and also any adverse events, in all the phases. So after phase 1 and phase 2, again the sponsor in industry, they have to set up a called end of phase 2 meeting. This is called end of phase 2 meeting. In the end of phase 2 meeting, whatever the information you gathered from the pre-IND, phase 1 and phase 2 complete, okay, one has to submit to the agency in the form of a module 1, 2, 3, 4. Okay, the clinical data will be always, should be submitted in the form of a module 5. The non-clinical part will be module 5. Module 4, and the drug substance and the drug product, they have to be in the module 3. Okay, so once the FDA says yes, this is okay, then you can proceed for the phase 3 studies. If not, okay, no, it cannot be done because the protocol is not acceptable, because the protocol is not containing the complete extensive data like inclusion criteria, exclusion criteria. I'll show what exactly it is actually going forward. Okay, I'm going to giving a whole picture here, and then phase 3. Once phase 3 is complete, then finally you're going to submit to the major NDA. NDA is a 505B1. 505B1 means is a pathway in FDA where you are going to submit new molecular entity, first drug substance. So during NDA, before NDA, one has to come up with the registration batches, 100,000 packable units. Okay, both drug substance also they have to meet the criteria of the requirement of ICH guidelines, and the drug product also they have to be manufactured 3 batches. Okay, 6-month stability, accelerated conditions of 40-75, and a long-term data minimum 12 months, and they have to submit. So by the time you come to NDA, all will be locked. Means that you have to establish the specifications, analytical method development and validation. What is the validation? Means that precision, accuracy, system suitability, all those things one has to come up, validate. You have to validate the process. Everything will be robust enough, established, and it should be locked. So once you submit the complete dossier to the NDA, if the agency says that yes, then you can go to the commercial marketing. Okay, till now, all these things will be called as a branded company. They enjoy the benefit of the patents. Okay, once after the branded company, once there are paragraph 1, 2, 3, 4. Paragraph 1 means no patents. Paragraph 2 and paragraph 3 means when the patents is expiry from the branded one, any generic industry can enter into the market. So here I am showing it here. Generic versus brand. Brand means till from IND to NDA. Generic means from NDA once after the patent is expired, or patent 4 is if when invalidate the patent, then generic company can enter into the market. Okay. Unlike an innovative company, generic company only needs to show the bioequivalence with the NDA. Generic company no need to follow the complete extensive clinical evaluation, whatever we did up to phase 1, phase 2, phase 3. They just do the bioequivalence only between the comparator between the RLD brand versus generic only. Okay. So an investigational new drug applications is a request for the authorization from FDA to administer an investigational drug or biological product to the humans. Okay, there are 2 types. One is IND, commercial IND, submitted mainly by the company seeking a marketing approval for the new drug. The second one is research, non-commercial, any research institution or any academic or any whatever sponsor is comes under the research IND. Okay, then the main important thing here is that the main 2 governing things are, one is ICH, International Conference for Harmonization. Every country will follow. So they have 4 things. One is quality guidelines. This is called whatever you manufacture the drug substance and the drug product, it will be covered under module 3, quality. The preclinical complete data will be, how exactly you need to present, what are the requirements you need to follow, the efficacy, mainly safety, preclinical safety. Safety is also involving the module 5. Okay, this is called, efficacy means on the human beings, how exactly the drug is efficacious, So mainly this is module 3, the drug substance, and preclinical will be mainly module 4, and all the clinical phase 1, phase 2, phase 3 will be module 5, efficacy. So this is the overall structure from the pathway from IND to NDA. So I'm going to show, just to simplify, CMC manufacturing drug substance and the drug product, non-clinical animal studies data, clinical, and you connect to regulatory agencies called health authority. Of course, quality assurance, I'm going to talk about what about quality. Quality will be there in the pharmaceutical industry, in the manufacturing as well as in the clinical. How they are going to monitor the, quality, risk-based approach, to make sure that everything will be aligned and compliance. So this is the overall tree for the ICH, The first module is non-ECTD. As I mentioned, is only administrative documents like Form 356, Form 1571, 1572, which I'm going to show. And the second module is quality overall summary. Okay. And the third module, what I mentioned, you need to submit all the documents to the FDA in the form of a module 3. like 32S means drug substance, 32P means drug product. And this is module 4 is nothing but our non-clinical, what I mentioned, module 4 data, and module 5 is a clinical data, complete like pharmacological data, then safety pharmacology, pharmacodynamics, and pharmacokinetics, like, population kinetics and everything, clinical study reports, everything will be. Remember, for the new NDA, it is about 10,000 to 20,000 pages when you submit to the agency. It's a whole bundle actually. You know that extensive data will be generated. Whatever you generate from the preclinical, whole thing will be captured, and while you're submitting to the agency for the at the end of the for the NDA. So this is very important. Okay, for every stage of phase 1, phase 2, phase 3, how you are going to submit, build the dossier package, the content, like module 1, cover letter, table of contents, form 1571 cover sheet, 3674 clinical trial certification, clinical supply labels, general investigation plan. So don't bother. I'm going to say what about all these things. I'm going to explain in the subsequent sections. Investigators brochure. This is nothing but a complete compilation of all the safety, everything from the preclinical to the NDA. Okay, module 1. Module 2, okay, don't bother about module 2 is nothing but the summary of module 3, nothing but the summary of module 4, nothing but the summary of module 5. All these three things will be captured in the module 2. So summary of CMC, whatever module 3, summary of module 4, toxicological studies, and summary of module 5, pharmacology and pharmacokinetics. So this is the whole gamut, the whole content, the whole dossier, what we file to the any agency, FDA or Health Canada. This is the typical format. Now I'm going to show the forms actually, So this is a 356 form, administrative document. So for the FDA, this is a typical 356 form. One has to file, while filing to the phase 1, phase 2, phase 3, whenever you make any amendments or changes, this is the document. Name of the applicant address, authorized US agent, product description, NDA, ANDA, BLA, biologic applications. If it is a supplement or any amendment or changes, establishment name, proprietary name, like application information, you have to click here, new drug application, abbreviated new drug application, biologics license application. So it's all typical fillable format. Or if it is NDA 505B1, what is 505B1? It is a new chemical entity, never before. What is 505B2? Already there is a existing NDA, but you are going to add a new strength or just any minor modification. So any change, whatever existing, like a new strength or additional strength or whatever line extension, it will be filed under 505B2. Okay. For biologics, the application type 351A, 351K. Okay. What is the submission type? So if it is a original application, you mentioned tick mark here, original. If it is a efficacy supplement, not the any toxicology supplement, post marketing, once you file NDA, when it goes to the commercial one, making any changes, post marketing requirements, post marketing commitments, any studies, say for example, in the case of recently vaccine, COVID vaccine has come to the market. There was no extensive safety and data, safety data was established because of the urgency, because of the pandemic, extensive emergency use of authorization, the vaccine was, the release to the market, the people were using. For the last 4 to 5 years, there was a tremendous, there's a lot of adverse reactions like, safety issues like clotting or whatever. So those times FDA will ask you, hey guys, this a lot of, adverse events, safety issues are coming up. You need to make a commitment to the post-marketing requirement. Can you please conduct a type of study to what extent the COVID vaccine is causing the clotting factor by animal study? What about pediatric study? Okay, you are not submitted any document to the pediatric study, like, only for adults after 18 years. So from the 12 years to 18 years adolescents, from 6 to 12 years is called pediatric studies. So all these things upfront at the phase 3, one has to make a protocol and submit to the agency and get it approved, how you are going to study. So not after the study. Okay. Likewise, REMS, risk evaluation mitigation supplement, human factors, annual report. Okay, those things which I'm going to explain what is annual report, all these things actually in the CMC supplement. So this is a form 356 typically, one has to submit. Then form 1572, the statement of investigator. Who is investigator? Any doctor, say for example, in Bapatla College of Pharmacy, say for example, Dr. Gopal Krishna, somebody is going to, conduct the study under his guidance. Then he has to submit his CV, his biodata, his qualifications, his certificate, because these are required to submit to the agency. For example, what is his name, address, city, educational qualifications, and he has to also mention a financial disclosure that I don't have any financial commitments, like name and address where the exact clinical study is going on, like, institutional review board number. So these are all fillable forms. Actually, one has to, fill in the administrative documents. Okay. Then sorry, 1 second. Okay, now, I'm going to explain what about clinical protocol. When I say that protocol is very, very important at phase 1, phase 2, phase 3, it has to be approved by the IRB, Institutional Review Board, okay, or IEC. In the Europe is called as IEC, Institutional Ethical Committee. So once this is done, you submit to the agency, right? So this is a typical clinical trial protocol. I am just showing this one because it's available in the public domain in the FDA by Boehringer Ingelheim company. So UDRA CT number, typically in the Europe they call UDRA number, EU trial number. So what is the trial name and what is the title? You're going to say this compound name, pharmacokinetics, immunogenicity, efficacy, and versus, all these things one has to, randomized double blind. Double blind means that, the drug will not be disclosed to either to the investigator or to the patient. Randomized means that, it cannot be, to prevent the bias, so the study will be under the randomized. And a parallel arm. Why it is a parallel? There are two things: what crossover technique and even parallel study, because it depends upon the type of the drug and half-life. Because of half-life is long, it cannot be done to the crossover. So parallel study. So the design of the protocol is very important in the, industry. The clinical research team, the clinical operations team, regulatory, they sit together, they develop the protocol. The design is very important. Okay. After the design, okay, this is a phase 3. Okay. Then comes to the inclusion criteria and exclusion criteria. These are very important. I'm going to show all the topics here: protocol date, trial name, how many number of subjects in the phase 3, 240 patients, then diagnosis, then main in exclusion criteria. Say, for example, they mentioned here, the trial will include male and female participants at least 18 years of age, who have diagnosis of the moderate to severe chronic plaque psoriasis, because this is the exclusion criteria. Those patients will not be involved in the subject, in this case, in the study, because the subject study itself to evaluate the psoriasis. People, so they have to, they cannot be included in the inclusion criteria. Likewise, what is the dose? What is the route of administration? So all these things will be captured extensively, how you are going to. This is nothing but, general investigational plan also in the phase 1 before NDA, phase 1 pre-NDA or even phase 1 NDA, IND, we have to submit to the agency. How you are going to submit? The protocol is the main part of phase 1, phase 2, phase 3, or even NDA. Anything if the FDA or any other health authority is not convinced, they will ask you to modify it. Okay. How many number of days you are going to study? 12 weeks, double blind, or 24 weeks? Informed consent. Whenever you are going to take the patient, you have to take a consent from them. Enrollment, right? So demographies. So all these things will be like what is the statistical model, PASI. So all these things will be captured in the clinical trial protocol. So the topics are medical drug profile, rational for each and every stage. You need to come up with what is the rationality, why you are going to the randomized and even double blind and everything. You need to come up with a justification called as a justification or rationality in the protocol. Any deviation from the protocol during the study, it has to be stopped and it has to be informed to the agency. If it is a significant variation, for example, if even a death occurs, okay, immediately you have to stop, inform to the agency, and the agency will ask you to modify, called a substantial amendment. Amendment and supplements are very important, in the regulatory filings, which I am going to talk in the subsequent sections. So while evaluating what are the primary endpoints, what are the secondary endpoints, just to brief outlay, say for example, we are going to evaluate any cancer drug. To what extent the cancer causing, there is a decrease in the cancer cell, the morphology, and to what extent the cell mitosis, there is a division or decrease in the cell, multiplication. So you have to come up with a kind of model called as a primary endpoints or secondary endpoints. You need to come up with, everything is a highly illustrative and justification, with a detailed, statement in the protocol. So this is a typical protocol any industry will follow. Any agency, any country, they follow critical protocol. Likewise, you have to also make a statistical design. What are the statistical, application plan? Which model you wanted to follow? Okay, there is a lot of statistical models. You need to display everything upfront to the agency, how you are going to carry out the clinical studies. Okay, then I'm going to show one thing like I think this much will be okay. I don't want to overburden you. Like this all clinical review, investigation, new drug applications, like pre-IND meetings, clinical trial protocol, which I have already explained, and classic PK studies and population PK is what is the difference between population PK and individual PK. So how over a period of subjects, a group of subjects, what is the PK data? What is the variance? So this complete whole gamut, at every stage, one has to, submit the data to the agency, and likewise bioavailability and bioequivalence, drug-drug interactions, okay, and then fixed dose clinical trials, titration with clinical trials, everything with placebo or any comparator, or if there is no placebo. So sometimes, the regulatory agencies that, once you develop the phase 1 studies, they justify that phase 2 is not required because it's a waste of time. We directly want to go to the phase 3. Why you want to go to phase 3? That's what here the regulatory agency, how to build up effective strategy on the disease specific. Okay, some people, they want to skip the phase 2. They will convince to the agency, hey, we have enough data. This is the data we have in extensive literature is available. Other countries also, they did it kind of study. We wanted to directly go to the phase 3 studies. So this is the kind of, how you are going to justify to the agents, at each and every stage, that is very important, okay. So as I mentioned, in the beginning of the things, like at every stage of phase 1 and phase 2, phase 3, there is a type B meetings, type A meeting, type B meeting, type C meeting, and a pre-NDA meeting. The meetings are only meant to negotiate with the FDA. If the FDA doesn't accept it, right, then you have to modify, and again you need to conduct the additional study, and then again you have to submit to the agency in the form of like, like module 1, 2, 3, 4, whatever I have mentioned earlier. So this is application. I'm just showing the example here. Center for Drug Evaluation Research for FDA. This is available in the public domain. Application number, administrative correspondence document, meeting minutes. Okay. This is a type B meeting, pre-NDA meeting. Okay. November 13, 2020. IND number. This is the drug substance name. Okay. Who are all the FDA attendees, respective department wise, and from sponsors, like from your college, if you are going to submit any application, who are all going to attend to the meeting, what is the background discussion. So that way, the meeting will be going on, and the FDA will keep lot of questions on the questions, and on that questions, the sponsor will give the answers, and again the FDA will respond to your questions actually. For example, they mentioned that without access to the batch data, okay, no, I'm not showing anything, but I wanted to show something which might be easy, okay. So the question is that on the target has been followed international conference on the harmonization, ICH Q3, in setting up the reporting of criteria for the impurities, okay, in the drug product. To be any impurity would be reported, okay. On the target, it include the associated chromatograms also will be supported. So they mentioned, does the division agree the impurity identified and the associated data will be adequate? The agency, yes, this approach is acceptable. So for all the questions, whatever the questions will be connected to the clinical, connected to the drug substance or the drug product, you need to list out all the questions, whatever you're going to ask, So some of the questions the FDA may ask yes, some questions may no. In the case of if FDA says no, again you need to make a justification, or you have to modify your protocol amendment or whatever. So this is the kind of approach continuously. Phase 1, phase 2, phase 3, type BA meeting, type B, type C meeting. The main difference between these meetings are only type B meeting will be elaborate one. Type D meeting will be only whenever you wanted to ask any one or two questions to the FDA, you can use the type D meeting only. Okay, this another type A, type B, C meeting, you need to make a briefing package. Okay, or 60 days before you need to inform to the agency. You have to submit to the briefing complete package what you are going to ask. You have to fix the date. And 1 week before you need to a kind of agreement if any questions. After the meeting, again you need to the FDA and the sponsor will sit together. They make a minutes of meeting on the type B. So these are the type A and type B, type C meetings in the FDA. Okay. Hope I covered everything, like how we negotiate in industry will continuous dialogue or communication with the FDA in the form of meetings, right from the IND phase 1, phase 2, and phase 3, like even after NDA also. Okay. Then, so this has been already covered from what is 57 and 1572. Okay, all these things actually. Then pharmacology, ICH M3, right? So now comes to the ICH guidelines. These are very, very important. One has to remember that, while chalking out the protocol or design or any developmental stage in the drug product manufacturing for the phase of module 3. So keep in mind about, the ICH guidelines. Q means everything quality, Q1, Q2, Q3, Q4, Q10. S means S1, S2, S3, S4. There is specific sections which I'm going to show here. The safety guidelines, carcinogenic studies, S1 to S1A to S1C. How you're going to develop the criteria, to develop the protocol, to incorporate the content as per this guidance documents. Genotoxicity in the preclinical, the criteria, the specific requirements can be mentioned in the ICH guidelines. Likewise, toxicokinetics and pharmacokinetics. What are those two? Because so how you are going to develop the toxicological studies? Again the same AUC at what levels of C maximum and T maximum. What is the kinetics and everything, like AUC, T maximum or C maximum, all other, So these things, how you're going to chalk out, what animal models you are going to chalk out, select strategy. That's where your regulatory strategy will be building, as per the specifications and the criteria enshrined in the, I'm sorry, in the safety guidelines. Toxicity testing, reproductive toxicology, biotechnological products, pharmacological studies, immunotoxicology, sorry, non-toxicological evaluation in the animal models, anti-cancer drugs. If you click here in the ICHN, you will see what are all the complete guidance documents. S1 means S1 to S13, etc. Safety guidelines. Safety means in the human beings is called pharmacovigilance, human beings. But in the preclinical, that is in the animals, toxicology. There are two things. All these things will be captured as per the safety guidelines. Likewise, again, safety. Efficacy. So efficacy, as you mentioned, that phase 3 is the main larger group of human beings, where to what extent the drug product is highly efficacious, the receptor protein binding, complete, the disease treatment, and with respect to food effect or pharmacokinetics, bioavailability, fasting, across all the subjects. And, these things will be carried out as per the efficacy. E means efficacy. E1, clinical safety and drug user in the long-term treatment. For example, efficacy, though you established, for example, for 1 year. But in the case of, say, for example, biologics, COVID vaccine, over a period of time, 3-4 years or even 10 years, continuous post-approval surveillance, they collect the data, safety to what extent, maybe it is a adverse reactions or adverse events. So all these things, which I'm going to show it in the subsequent, sections. So clinical study reports, this is very, very important. As I mentioned in the earlier, once you develop the protocol, once the study is done, okay, in the clinical sites, okay, you do the data lock, you do the statistical evaluation, statistical analytical plan, SAP, and then you develop the clinical individual reports, summary reports, everything will be captured how you are going to do it in the E3. Likewise, dose response studies, ethnic factors, good clinical practice, clinical trials in geriatric population. For example, in the case of geriatric population, there are special considerations. We cannot not like a adult population, like 18 years to 15 years or 60 years. For example, geriatric population, what are the main considerations? Their biology, like particularly in the case of dementia, Alzheimer's disease, like while doing the development of the anti-depressant drugs for the adults and for the geriatrics, you need to consider the dosing regimen, the geriatric population, keeping in the perspective. Likewise, general considerations for the clinical trials, statistical principle. This is very, very important. Upfront with the protocol, one has to get approved by the agency on the statistical analytical plan called SAT, statistical analytical plan, SAP. That is very important. If FDA may ask you this protocol is not acceptable, you need to modify, amend the protocol, amend the statistical analytical plan. So that means that in the course of the NDA, at every phase 1, phase 2, phase 3, you need to have whatever you gathered so far the data show to the agency, and whatever you are going to propose the protocol design show to the agency, get it approval, then only you need to start the clinical study. So next, efficacy, same thing, pharmacovigilance, clinical study reports, all these things, pharmacokinetic study, food effect, fast effect, how many number of, complete design will be enshrined and enunciated in the, protocol. Likewise, multi. So with this, I think next, quality, CMC. So far we have discussed about non-clinical and clinical. Now there is another dimension called as a drug substance and drug product. If you talk about a tablet, particularly in the case of pre-IND, clinical, non-clinical phase of toxicology, just only the practice that take the drug as such dissolve in the water or whatever suspension and just to administer to the animals. As you evolve to the phase 1, phase 2, you try to kind of come up with a tablet or capsule, so you make a drug product. The drug substance is the active pharmaceutical ingredient. The drug product is the combination of the excipients. I'm talking about the small molecules. For the biologics, sometimes the drug substance is the same drug product also when you take into the medical devices, combined together. For example, in a in a ampoule or like IV, IV like vials, the drug substance in a vial is a drug product, but the drug substance in the solution is nothing but a drug substance. So all these things will be module 3. CMC means module 3. How exactly you need to develop, we have to follow the ICH guidelines Q1A, Q2 analytical validation. Impurities are very important on the stability, pharmacopoeias, biotechnology specifications, which are very critical. You need to come up with a kind of acceptance criteria, manufacturing practice, pharmaceutical development, Q8, risk management, quality system. All this Q means quality. Okay. Now I'm going to show the quality guidelines. Okay. This is particularly I'm going to capture. The CMC content will be captured in the module 3 of the ECTD, and as per the ICH guidelines is quality. For example, I'm showing it here. Formulation development. Okay. In the initial formulation development, say for example, if this is the formula composition, but here in the main content in the in the developmental phase QBD, quality by design, risk-based approach. If I say that 13% of methoxyl K4M is really working well, meeting the dissolution criteria called CQA, critical quality attributes. The final drug product, CQA, blend uniformity, dissolution, assay, impurities. When you say that 13% is really working well, this is the formula composition. What will happen when you keep 8% low? What will happen when you keep 18%? This is called a design space. Okay. So during the course of the development, one has to come up with the optimization of the formula composition ingredients, excipients called as material attributes, their risk assessment, and what is the process. Say for example, when you blend the whole direct blending process, when you adopt, I am blending only 10 minutes, is coming well for blend uniformity. But when you blend for 5 minutes or 15 minutes, varying conditions of blending time, so called process, process optimization or process changes. So the material attributes, process attributes together will constitute the overall quality of the drug product. The quality by design is inbuilt, not at the end of the manufacturing of the quality or tablet or capsule. So during the initial stages of the developmental process, product and process will be developed and optimized. But when it comes to the stage 2, process validation, as I mentioned, that once you submit NDA, before going to the market, commercial market, the companies will manufacture 3 validation batches as per the commercial requirement batch size. Okay. In the R&D formulation stage or development stage, only small equipments are used. When you go to the scale-up level, bigger equipments, bigger granulators, bigger blenders. So what will happen? You need to come up with one has to establish the robust process parameters, okay, robust criteria, specifications, at stage 2 of validation. Stage 1 QBD, stage 2 is risk evaluation, risk-based approach of process. Okay, then commercial manufacturing. Even the commercial, during the commercial manufacturing, every 10 batches or 100 batches, if the quality is good, if any issues are coming up in the process or in the product, blend uniformity failing or content uniformity failing. So one has to keep everything in the data in the form of a QSMAR, quality, management review, like, quality assurance will keep all the data actually. That is the in-process controls, release controls, final release specifications, even post surveillance, post commercial manufacturing also. This is the whole, this is the typical diagram in the QVD. Okay, as I mentioned, this is the APA particle size. When you select the APA particle size, when you say the risk is low, why it is low, one has to make a justification. Say, for example, medium. Content uniformity is a critical quality attribute. APA particle size, medium. Why? If it is high, say for example, why? So you have to make first of all design a QBT design in the form of this called a risk table, both for excipients and the critical quality attributes, and then conduct the studies DOE, design of experimental batches, then make a conclusion out of it. Okay, fine. So this is the overall developmental of CMC, formulation development, optimization, and then final stage to the validation, submit to the AGG. So we have covered the clinical phase protocol and also the CMC manufacturing guidelines. Now, comparison between the IND, NDA, BLA, biologic application, ANDA. IND, NDA, innovator and brand molecules. Biologics are biotechnological, manufacturing sponsored companies. AND is abbreviated, called as a, even once the safety is completely established, they call OTC, just on the counter products. Okay, this is the pathway, IND, NDA, BLA. So IND, typically we follow investigator sponsored or emergency, like, preclinical data, CMC, as I mentioned, the CT protocol. NDA, 505B1, 505B2. As I mentioned, 505B1 is a new chemical entity, hitherto not find out any, it's completely new molecule. Okay, there you develop the drug substance, you develop the drug product, comes under the B1. Whatever the existing already, you made some modifications on the line extension or lower or higher dosage form, a kind of stuff, you do 505B2. Biologic applications, 351A and ANDA, generic or biosimilar. Biosimilar is nothing but a bridging between the innovator branded company biologics versus the biosimilars, called as 351K. So again, comparison of the initial regulatory applications, in USA, as I mentioned, is IND, investigational new drug applications, IND. But in Europe, they call CTA. Other countries also, like Canada and I think even China, they call it CTA. CTA means clinical trial applications. They apply using IMPD, investigational medicinal product dossier. Okay, this is called ECTD format. Likewise, UK CTA. So it's just only the naming will be different. The more or less the content will be the same, but you need to know how exactly you need to compile the data, the dossier in the form of a specific country requirement. Country to country is, it varies. Okay. Likewise, CMC module, full ECTD module 3, in the case of USA, full IMPD section 2. Non-clinical module 2, module 2, 6 and 4. Clinical. So these are the main differences actually, which I don't want to go deep, because mainly the content will be the same, but we need to fine tune and modify as per the country specific requirements. For example, China, it may need more kind of, batch records, chromatograms, even the clinical studies, whatever you do submitted and approved in USA and Canada, the Chinese health authorities, it may not accept, because they may ask you to conduct some of the clinical studies on the mainland of China also. So that's why how you are going to build up the regulatory strategy. First of all, what I said in my previous company, so what we do that pre-clinical studies are before opening an IND to FDA, we do the quality overall summary and conduct some trial batches in Canada. because the Canada path in some cases is very easy, and get the data, gather the data. The data you submit in USA in the form of a pre-IND application. It's a kind of regulatory strategy. Some countries, once you develop and submit the same document, whenever you're going to, to some extent, you can derive the data and submit to the other countries, wherever acceptable and, wherever it is agreeable actually. So this way we need to country specific. Likewise, whenever you are going to make changes, like, as I mentioned, the amendment, and I'm going to talk maybe in the subsequent chapters actually, okay? So this is very important, change controls, change management is very important in the pharmaceutical industry. Maybe it is a clinical amendments, maybe it is a CMC amendment. What is amendment and what is not amendment? An amendment is a major change. Major changes are, first of all, you need to submit, hold for 30 days before getting approval, okay, from FDA. Okay, if the changes are very minor, no need to inform to FDA. Just to go ahead and submit in the annual report. Okay, there are two things: major changes, minor changes in the case of phase 1, phase 2, phase 3. But in the case of post-approval changes, there are 3 change controls. I mean, one is major change, the second one is moderate change, the third one is annual report. If it is a major change, that means how you are going to determine whether it is a major, minor, or moderate. Say, for example, you are going to add a new site. Okay, one company is already commercialized a new product, it is in the market, but when you want to add a new manufacturing site to the existing site, you are going to add a new manufacturing site because, say, for example, Bapatla College Pharmacy is a new company. They have already developed a product into the commercial market, but they wanted to hire another college as a new additional site because of the expansion. So that case, you need to evaluate. Okay, if it is a modified release dosage form, definitely it is a prior approval supplement, because modified release, sustained release molecule, as per the guidance document, you need to have a bio study whenever you wanted to site transfer to the another manufacturing site. Whenever you wanted a new site, which is already GMP approved one, audited one, okay, because of modified and sustained release, you cannot do the simply like this, in the second site, is a major change. Likewise, whenever you wanted to delete a blend uniformity which is already approved, okay, in the commercial manufacturing process, that is a major change. But whenever you wanted to use a minor screen in order to delump the granules, it is a minor change. No need to inform to FDA. Just go ahead and report it in the annual report. So there is a complete criteria, the impact assessment called regulatory impact assessment, what we do in the form of a change control system using a Viva Vault, okay, for the clinical domain as well as in the manufacturing process. This way what we do, okay. So I mentioned here amendments here, initial IND, okay. What are IRs? IRs are called information requests. Say, for example, whenever you want to submit any submission dossier to the agency is not able to locate any information. Hey guys, can you please let me know where are the batch records? Oh, I forgot, I'm going to send it to you. Can you send it to me within 1 or 2 weeks? These are called as information amendments or information requests. These are very simple, but major amendments are only you need to wait for 30 days. A clinical hold, okay, please don't conduct study, wait for 30 days, okay, then we'll come back, called as amendments, okay. So during the phase 1, phase 2, phase 3, IND and information requests, and post marketing, the supplements are called as a CB30, and PAS, prior approval supplement, is a major change because the FDA will inspect, you have to wait for 4 months. Annual report, no need to wait, just to implement. After 1 year cycle, you just file to the agency. If it is CB30 moderate change, first inform to the agency, wait for 30 days called CVE 30. Then if you don't hear anything back from the agency, then you implement the changes and then you go ahead. So these are called as change management, change controls, what we do it in the commercial domain, I mean manufacturing CMC domain as well as in the clinical domain. Regulatory impact assessment as well as a quality impact assessment, we do this way. Knowing about the ICH guidelines, knowing about the country specific guidelines, knowing about to what extent the change will be impacting on the significant or non-significant or the critical quality attributes of the product or on the clinical, impurity profile is very important, For example, if any degradant is causing any impurity, you need to conduct, qualify the impurities as per the ICH limits. So this is the overall CMC cycle. Okay. So again, it's a repetitive type A, type C meetings with the FDA. Now I'm going to show to the demo to the change management, change controls. Okay. Major change, moderate or minor change. How in the industry, typically, each department will assign the task to the different people. They do their impact assessment. I'm going to show it in the demo. So this is a typical Viva Vault, typical Viva Vault, affiliate, home, library, health authority interactions, registrations, what registrations is USA, Europe or Canada, applications, products, reports, Viva. So this is a template actually Viva. Viva RIM means Regulatory Information Management, RM. For the quality is called as Viva Vault, okay, QMS. Quality management system. In the Viva, it's a customized tool actually. So whenever you do the investigational application name, these are all self explained and is all customized. One can it's a very easy. The application details, publishing details, regulatory objective is very important. Submissions, therapeutic indications, countries, product families, registrations, correspondences with the agency. So then Then health authority questions. When you come back, you need to track it here. It's a kind of tracking mechanism. Likewise, whenever you want to make any change controls, also you do the similar way. Commitments, whenever you want to make any commitment to the agency that okay, this I think as of now we are going to study the juvenile animal study because we don't have any data to replicate the pediatric study, the things we are going to commit in the future juvenile animal study in order to correlate the data to the pediatric study in the future. We have to make a commitment in advance. Okay, these are all the regulatory applications, system information, all these are self, home, affiliate name, library, health authority. When you click here, all drop down menu, you will see all the features. You see, investigational submission 007 sequence protocol amendment, dossier format, ECTD, submission type, okay, application type, planned submission date, actual submission date, publishing details, dossier details. So everything is very easy. Viva Vault is really one of the best tool one has to acquaint, very well in order to know the complete regulatory as well as quality. Say for example, IND 775577 protocol amendment, the form 1571, cover letters and USA clinical study reports. Everything is self-explanatory. This is called Viva Vault. See. All these are historical sequences actually. Whenever you submit one version now, after 2, 3, 4 versions, whenever you make any changes in the protocol amendments, then you have to assign a sequence 005, sequence 006, 007. Okay, this is a typical format of Viva Vault for the regulatory information management. It helps not only in the compilation of the all the module 1, 2, 3, 4, but also you can review, but also change controls mechanism and review, change controls impact assessment, or before starting the change controls, whenever you wanted to assign the task to the different departments to know their impact assessment, like quality assurance, R&D formulation, commercial, regulatory, legal. So anything you can assign a task, child action, whatever. Okay, this is a typical one. So this is the Vault RIM, regulatory information management. This is complete the dossier details, validation results, all validation data, submission administrative information, regulatory objective. So the regulatory people have a lot of departments within the regulatory. People will do the strategy, one department. People will do the regulatory operations, that is one. People will do the regulatory labeling, that is one. People will do the regulatory submission management, one. People will do the health authority questions, how they interact, and there's one. There are so many departments within the regulatory, within the clinical, within the QA. Please let me know if if I'm able to, if any chat box also, please let me know your questions and your ideas where I can capture and I can even address all those things while the session is going on. Okay. Sorry, 1 second. Let me cover my this one. Are able to from the current one. Okay. Then comes to this one is very easy. Say for example, as I mentioned, change control and change management, regulatory impact assessment during the NDA phase 1, phase 2, phase 3, even after NDA. Change management is one of the important. Whatever you submit to the agency, you need to follow the same thing, batch record or protocol, any process. If you wanted to make any changes in the next study, right, you have to evaluate the impact assessment. Like, post approvals, once after approval, commercial market, major change. Major. A major change is a prior approval supplement. That is minimum, it may take 4 months to 5 months, you have to wait. In Europe, the major change is called as a type 2. In USA, it's called as a PAS, prior approval supplement. In Europe, it's called as a type 2 variation. Okay. So that means, first of all, tell and wait. Tell to the agency because it's a major change, because you did the impact assessment, because it's a major change. Why it is a major change, you need to know. A regulatory people and quality, they have to evaluate with respect to the ECTD guidelines, quality guidelines, impact on the quality, clinical, you have to come up with a measure, right? Then it's a type 2. Tell, wait. Then only you have to implement the changes. Because of this one is a significant impact. If it is a moderate change, changes being affected, CBE 30. CBE 30 in USA is equivalent to type 1B variation because it's not a major, it's not a minor, it's a moderate. Say for example, bulk hold time study. Bulk hold time study means that whenever you wanted to film coated tablets before packaging, you wanted to extend the shelf life of the bulk coated tablets. Okay. With the approved protocol already we mentioned in the NDA dossier, we wanted to extend the data from 18 months to 24 months or 24 months to 36 months. Okay, you have to evaluate bulk hold time studies in process 1. Already we mentioned to the FDA in the last time in the NDA, we made a post-approval commitment. Okay, just we are going to update the bulk hold time study of the film coated tablets prior to the packaging into the bottles, with the real-time data, whatever we generated at our facility, it is not a major change, it is a minor change. So what you do, tell and do. Tell, wait and do. First of all, you inform to the agency means the tell, wait for 30 days. If you don't hear back anything from agency, then you can implement the change. Yes, extend the shelf life and go ahead, called a moderate impact. Minor impact means that, say for example, you are revising a batch record. The batch records already submitted to the agency, but since because you are changing a little formatting, right? These are not very major or minor. So whenever the audits are coming to the on-site, you can show the batch records. These are very minor, so no need to make any changes. So these are minor. So these will be captured in the next annual report cycle. So there is a cycle every year. You need to update what are all the changes in the last 1 year. Say for example, Babatlagaja Farms has a commercialized one market today. Next year in the same date, during the course of 1 year cycle, you need to sum up all the prior approval changes, all the moderate changes, all the change controls, all the minor changes, make a list of changes and everything, called as a 1.13 module 1.13. Okay, 1.13 is nothing but a summary of manufacturing changes. So the respective modules you need to update. So it's called as how. So in Europe it's called as 1A. So country specific guidelines are different. USA comparison with the USA and other things and all. Then the main thing is pharmacovigilance. What is pharmacovigilance? Safety. What is safety? What is safety? I want to simplify the diagram. As I mentioned that in the case of safety guidelines is there as per ICH guidelines, right? Pre-IND animal toxicological studies you will evaluate toxic or not. But there you derive the data to some extent. But when the studies are going on in the progress like phase 1, phase 2, phase 3, in the human beings, safety data will be updated periodically. Okay, they called DSCR, called as there are two things. During the developmental stage of phase 1, phase 2, phase 3 and up to the NDA, they are called as a developmental of safety update. Okay, periodically. 60 days, I think definitely when the data lock is there to that particular safety issue, signal, you find out the signal. From the date, immediately 70 days, you need to submit, inform to the agency. But then onwards, annual update. So sometimes whenever in the case of NDA, once you submit post approvals, 4-month update, safety update. So safety updates are very periodically. If it is very lethal, if any person died, immediately within I think 7 days, you need to report to the agency. So 7-day period, 15-day period, 60-day period, quarterly, okay, then annually, then after that every biannually. So there is a periodic update of the safety report. Okay, how to classify the pharmacovigilance criteria based upon the intensity of the adverse reactions. Safety suspected adverse reactions, suspected unexpected adverse reactions, adverse events. Okay, they classify every time you need to. There is a pharmacovigilance cell department within the clinical team. They do the GEM software or SAS or Excel sheet. They find out, they will, they collect the data. So periodic benefit risk evaluation report, paper. Okay, this will be at the post to NDA, and periodic safety update reports, PSVR. Also, maybe I think there are different names, after NDA post approvals, even post approval and commercial market, whenever there are some vigilance adverse reactions or adverse events, they they file to the agency, they collect the data in the form of a PBR and PCR. DSVR means that during the developmental stage of phase 1, phase 2, phase 3, collect the adverse reactions or adverse events, make a report and submit to the agency. DSVR. So all these are, the a kind of, safety update, depending upon the intensity of the adverse reactions, adverse events. Quarterly update, and I mean, there's a criteria in the FDA as well as in the ICH guidelines. This is very, very important, pharmacovigilance. So pharmacovigilance also, there are how exactly you need to make a report as per ICH guidance document, as I mentioned in the safety guidance documents, and country specific guidance documents also you need to follow, make a criteria, collect the data, periodically you update the report to the agency. If it is lethal, then immediately you have to stop clinically, hold it, clinical hold, don't conduct any study, wait for the investigation, just to wait for to hear back from the agency. Okay, annual safety report, annual investigational new drug safety report, expert reports from the renewals, addendum to clinical overview. So this is overall pharmacovigilance update. So now clinical operations. So many people in the pharmaceutical field or chemistry people, what we think that, working in a tablet manufacturing or quality assurance or, a kind of generic industry or innovative industries only, but there is another dimension called as a clinical field. So clinical is very important. One, you understand the clinical protocol design, what I mentioned in the earlier case. So right from the clinical research department, clinical operations and clinical review, clinical investigational, there are so many departments. First of all, site, identify the site where you are going to conduct the study. Who is the investigator? Okay, what are his qualifications? Whether the site is approved or not. Site activation, then patient recruitment and enrollment. Take the informed consent from the patients. Okay, whether the patients who are all being enrolled, whether they met the criteria of the inclusion criteria, exclusion criteria mentioned in the protocol. So the investigator, the team, they have to assess all these things. Okay, so site management, patient recruitment, clinical supply management, you need to ship the samples from the your manufacturing unit to the clinical site. Record management. Okay, once you conduct the study for 1 month, 3 months, 6 months, over a period of time, you monitor the clinical studies, see if any adverse, events or if there is any protocol deviation during the course of the clinical trials conduction of the studies, then you have to maintain all the records. Okay, clinical safety monitoring and the pharmacovigilance activities, logistics, data management. Once all the clinical studies are over, the last subject, last visit, data lock, they call it as DL. Okay, once the data lock is done, then before making any clinical study report, CSR, they do the statistical analysis. It's a complete analysis actually. Then they do the clinical study reports. Those reports, they submit to the agency. So how the reports like this, I think maybe I can show it in the one document, baseline document, there's a lot of, individual data, then compiled data summary. That's why I mentioned that, once you submit to the agency NDA document, 10,000 pages, not even 10,000. If you see maybe 20,000 or 30,000 pages, there is a third party site. We give it to them. They keep on publishing and submitting to the agency. So all batch records, all clinical study reports, TLF, total list of figures, ISS and ISE, integrated summary of safety data, integrated summary of efficacy, ISC data. There are so many terminology in the clinical field, all these are mentioned and compiled in the module 4 and 5. Module 4 is a non-clinical, again module 5 is a clinical, module 3 is a CMC. The summary of module 3, 4, 5 and will be in the module 2. It's very simple. Okay. Then I think this already I have discussed. I think I don't want to discuss more because China clinical trial application, CTA and IND both are same. That's it. In Europe called as IMPD. Okay. Then thank you so much for the opportunity. And with this, if any questions, you can email me to my email ID rbrao9@gmail.com, or you can keep it in the chat box or in the to the Bapatla College of Pharmacy. They can give it to me. I can keep it back to that, in the interest of time, so that I have covered everything. So before that, I wanted to. So now you tell me whole 30 days class or whatever, the complete I have covered everything. So pre-IND, IND, phase 1, phase 2, phase 3, all these are clinical, both CMC and clinical, NDA, innovator company, and then I'm sorry, innovator branded, pharmaceuticals. Then after post NDA, again there is a called paragraph 1, 2, 3, 4. If anybody wants to invalidate the patent, or if they want to file or develop any product after the patent is expired, they called as a generic market. So that's why how the generic market, the generic industries are branded industries, the complete overview right from the drug discovery, to identify the drug discovery, animal studies, all the phases to the NDA, post NDA. Okay, then they enjoy the benefits of the NDA for a period of patent exclusivity for maybe 15 or 20 years. Nobody can manufacture the same drug in the same pathway unless they invalidate the patent. Hope I have covered everything. This is the overall regulatory pathway. It's very simple. You have to know the module 1, 2, 3, 4, ICH guidelines, individual specific regulatory country guidelines, variations or change controls, change management, impact assessment, and quality impact assessment. These are the main highlights which I wanted to share with you. Hope I have covered the real practical industry perspective to the to the people who are already watching, to the budding students of pharmaceutical field as well as in the industry. And please keep me posted rbrao9@gmail.com or to the Bapat College of Pharmacy if any questions. Thank you once again to the management and to the respected principal, Dr. Gopal Krishnamurthy, who is the pioneer of pharmaceutical movement in India, because I'm seeing a lot of his patents and research pursuits. Thank you so much.
Any questions from the audience?
Yes, ma'am. I posted the question in chat box, ma'am.
Sir, here it is a question, sir. IND and NDA process can fast up by using AI technology.
That's correct, because it's evolving, because there's a called as a real world data, real world of evidence. Whatever the data from the different agencies like clinical supplies and hospitals and say for example pharmacies and clinical settings, they collect the data called as recently real world data, real world of evidence. For example, whenever you don't have anything like AI can be used similar to that kind of, actually it's evolving actually still, but still whenever there is a specific disease oriented, to some extent we can use the AI, but not completely. But in the form of a real world data, real world of evidence, we can use the artificial intelligence. That's correct.
Any questions from the students' side?
In the interest of time, maybe ask them, collect all the questions in the bundle, everything. So you can keep me posted also.
Okay, sir.
Yeah.
Ma'am, I posted another question in the post chat box.
Yeah, you mentioned that IND and NDA process application fee details, sir. Hello?
Sir?
That is your question actually?
Yes, sir.
Yeah, that is millions. That is millions. Even for NDA, for the NDA, uh, uh, it is one of the important point actually. It's actually millions. To file an NDA in the case of FDA, they charge more amount. That's why you have to make sure that all the modules, module 1, 2, 3, 4, there are no incomplete information. Everything should be complete. Okay. Otherwise, they give you a refuse to receive. Refuse to receive means they will not give money. They just to keep some money. They just give only nominal money. All waste of money. So it's all maybe 1 or 2 millions. So it's all billion cost whenever you file NDA. That is why regulatory strategy building is very important right from the beginning of the pre-NDA, NDA, I mean IND phase 1, phase 2, phase 3. Say for example, whenever you miss one document, right? For example, if you don't capture the data from the preclinical, if you don't bridge the gap between the impurity profile comparison between the non-clinical batches versus the proposed commercial, it is a refuse to receive. You're losing a lot of money. I think there's a lot of resound, I think.
Ma'am, another question, ma'am.
Wait, ma, wait. Okay,
Yeah, I
okay.
think the next question, you can speed up the process by paying extra fees. No, no, no. Actually, there are two things actually. The only thing is that, fee is limited, but it depends upon the specific product. But only thing is that by just, okay, I think there are two things. One more, this is very important. Not the fees. There is a priority review. There is a standard review. There is a expedited review. Okay. So do you understand? Standard review, whenever you file NDA, they may take around uh, 12 months or 8 months like this. But in the case of priority review, right? Priority review, then they will do within a span of 6 months. 6 months review, because you need to also justify. You make a request to the agency that why you are going for the priority review, not the standard review. So because of unmet needs, okay, this is very important actually. The drugs has been classified as rare diseases, right? Orphan drugs, okay, under unmet needs. Because I'm going to submit an NDA, which is very typical one. So far, all the existing drugs are not able to meet the disease conditions. My drug is able to meet unmet needs. That's why I'm going to file under priority review. In that condition, you can expedite the review process.
Thank you, sir.
Thank you.
Any queries from the student side? Thank you, sir. Sir, we are sharing your certificate also, sir.
Thank you, ma'am.
Thank you, Dr. Bhaskara Garu, for sharing your valuable experience, expertise, so that the students can benefit not only from theoretical perspective, so that now, after listening your valuable lecture, the students might be in a position to file practically also. So it is really useful, and as a head of the institution, I congratulate and I appreciate your sincere efforts, and I hope your cooperation and support in future also, so that such kind of deliberations are required to strengthen the quality of students' education in particular. So I'm wholeheartedly appreciate for your commitment to support the student community. Thank you.
Thank you so much, Dr. Principal. I really appreciate your thanks and sincere efforts. Thank you so much. I appreciate it. Yep.
So the participants can fill the feedback link shared in the YouTube chat box, and please fill it so that within short period of time you can get the certificates. So thank you all participants for your enthusiasm to involve, to participate, to learn new things, and I hope such kind of participation will encourage the organizers to involve, to conduct such kind of deliberations with expertised peoples like Dr. Bhaskar Garu. Thank you, one and all.
Thank you, sir.
Okay, bye.
Bye, sir. Bye, sir.
See you.
See. Thank you.
మాది ట్రాన్స్పోర్ట్ అండ్ శ్యామ్ ఉన్నాడు సార్ శ్రీరామనే అన్నయ్య నుంచి తీసుకున్నాను చెప్పాను డీటెయిల్స్ అన్నీ చెప్తే సరే.
Participants, please fill the feedback form, and it is also posted in the YouTube also. So kindly please fill and get the e-certificate. So we will directly send the e-certificates to your mail IDs. Thank you. హలో చేయలేదా? నేను సెండ్ చేశానే. అవునా? ఓకే, ఒక్క నిమిషం ఆగండి, నేను పెడతా. ఓకే, నా దాని నుంచి పెట్టానా యూట్యూబ్? సార్ ఒక్క నిమిషం సార్ మాట్లాడు NDA చేస్తాను.